Good afternoon and good morning to the attendees from the US. My name is Loic and I'm the co-founder and CTO at Atinary Technologies. I'm thrilled to welcome you all to our webinar today. We have an exciting topic to explore, closing the loop in research and development by combining AI and automation to accelerate the optimization of chemical synthesis. I'm honored to introduce today's guest, Oliver Schilter, PhD candidate and researcher at IBM Research in Zurich, and Daniel Pacheco, ML engineer at Atinary Technologies. Today, Oliver and Daniel will share with us how combining AI, robotics, and data analytics through Atinary SD Labs and IBM RoboRxN allowed them to expedite reaction scouting and optimization. The focus is on iodination, a class of reaction that enables the synthesis of complex organic molecules. The work that we'll present here was recently published in Chemical Science from the Royal Society of Chemistry. Before we get started, a few ground rules. The webinar is being recorded and it will be made available on Atinary's YouTube channel shortly after the webinar. Everyone is on mute except for the presenters. And last but not least, I'd like to draw your attention to the toolbar at the bottom of your screen. You will find a Q&A module to type in your questions. We will monitor the chat throughout the presentation and will direct your questions to our speakers at the end of their presentation. Without any further ado, Oliver, the floor is all yours.
Thanks, Loic, for the introduction. As Loic mentioned before, today's talk is about combining Bayesian optimization and automation to simultaneously optimize reaction condition and the routes. This work started roughly 2 years ago as a collaboration between Atinary and IBM Research, and recently got published in Chemical Science, as Loic mentioned before, a couple months ago. And in the end, we will share a QR code for the publication where you can have a read in more detail later on. So further ado, I quickly introduce myself and Daniel. So I'm Oliver Schilter, as Loic mentioned before. I have a bachelor and master in chemical engineering from ETH Zurich. I then got an introduction into machine learning while doing my master thesis at MIT in the Jensen lab by learning how to predict the yields for reactions used for building up DNA-encoded libraries. And then did some internships in the pharmaceutical world at Roche and CSL Behring before I joined as a PhD candidate at IBM Research together with EPFL in the LIA group by Professor Schwaller. On the left here, you see actually an image from myself in the mountains starting one of the reactions. Yeah?
I'm sorry to interrupt, but we don't see your slides.
Oh, sorry. You see them now?
Yes, now we see them. Yes.
Sorry. So I go back again. But anyway, the introduction of myself is not so important. So I did an internship in Roche and my PhD at IBM Research and together with EPFL. And here on the left, you actually see myself sitting in the mountains running one of the reactions that we are talking about. This is the cool thing about a cloud-powered lab, that you can run reactions from wherever you want. So Daniel, my co-presenter, will introduce himself.
Hi, my name is Daniel Pacheco. I am a machine learning engineer at Atinary Technologies. For my background, I studied chemical engineering at McGill University in Montreal in Canada. And then I came here to Switzerland to study my master's at EPFL in computational science and engineering. And ever since, I joined Atinary as a software engineer originally. And now I am a machine learning engineer for a bit less than 3 years.
So thanks for your introduction. So let's have a quick word about how we can leverage AI to accelerate R&D. So in general, R&D is in a cycle where you usually start with a hypothesis, then you do an experimentation to confirm or deny your hypothesis by doing, after the experiment, you usually do analysis to see if actually the outcome is the one that you wanted. Then you report, you question, you study, and you form a new hypothesis. This cycle can be aided with AI in several steps. For example, in hypothesis generation, we can use generative AI to come up with new molecular structures that are most likely drug active and so on. Then we do confirm it via experiments, and even in the analysis part, AI can help us, for example, to use structural elucidation combined with AI to find out if we actually did the molecule that we wanted, and so on. In today's talk, we mainly focus on how we use AI in the experimental part, so how to suggest new reaction conditions with the help of AI and Bayesian optimization.
So here is the closed loop depicting how users can leverage SD Labs to further accelerate this research and development process. So typically the first step, step number 1, is where the users designs the experiment. So here they would design the search space, the objectives. This was a joint work with IBM where we were able to select all the parameters that we wanted to optimize. The second step is where the AI algorithms will then take this parameter space and then recommend experiments to carry out. Step 3 is where the user, in this case, RoboRxN, the robot, would take those recommendations and carry out the experiments, yielding some results, which are then collected at step 4 to build digital libraries such that users can visualize this data under the data analytics platform. They can gain insights of how their experiments turned out of the Bayesian optimization campaign. And also this information can be leveraged to generate new recommendations in a new iteration to continue a closed loop optimization, accelerating R&D.
Thanks, Daniel, for this quick introduction. So let's have a quick look at the agenda. So we already talked about the motivation. What we actually want to achieve is closing this loop. And then the next question is how we combine this robotic platform with the optimizer and which reaction we want to actually optimize for, and so on. Then some problems we face along the way. So you can maybe learn for your own projects and own optimization campaign what to focus on, and so on. Then the results of our optimization campaign, some insights we gained there, and as well, we end with a question and answering session in the end. So the status quo is when we started the project is we already had a robot that was connected to the web through a user interface. This was already done. And on the other hand, Atinary already had their optimizers that could be queried via API to get new reaction parameters. But the question was, how do we actually connect these two? So this, and after we connected them, what reaction we want actually to run to see to prove the concept. So first, let's have a look at our robot that we have here at IBM Research. It's a ChemSpix Flex unit. It has 6 reactors that each contain a volume of 150 milliliters that are stirred and can be temperature controlled and cooled. Even we can even run reaction in inert atmosphere. We have one robotic arm that can either attach a liquid handler, so we can dispense liquids, or we have one solid dispenser, which dispenses little glass ampules of solids that then are crushed in the reactor, so the solids are dispensed. We also can use analytics after the reaction to see if we actually formed the molecule we wanted or determine the yield of the reaction. We do this by injecting a little sample of the reactor into an HPLC that splits up the different components in the reactor, and then we use analytics measurements like mass and diode array detector to see which substance it is. So we can store roughly 30 liquids in the robot, so we can have solutions or pure liquids. We have 120 positions for solid ampules, and usually, as mentioned, it's controlled through a web interface because it was originally developed for retrosynthesis. For example, here you see a retrosynthesis of the molecule aspirin, where you would the tool suggested to do the Fischer-Tropsch esterification. So we can actually, if we press run, this would then send a command onto the robot that actually runs this reaction. But we didn't want to do explorative science, but we wanted to do reaction optimization, so we had to adapt this tool a bit. So which reaction we selected to do this is the synthesis of iodoalkenes. So where we replace a terminal hydrogen from alkene with an iodine group. This is usually done by some iodine source, so iodine salts like sodium, iodine on the reagent. Then as normal reactions are usually run in a solvent, so some solvent and a temperature. So we run it at room temperature or high temperature and so on. Some application of these iodoalkenes are, for example, we can form dienes, which this reaction reported here in literature, as well as we can do some cycloadditions to building furans, which is reported here, as well as we can also form other cycloadditions to form hydrogenated reactions, so halogenated agents. So there's a variety of applications for this kind of iodoalkenes. So we looked at how we can form these iodoalkenes. So we selected two routes. One is using NES as an iodine source as well as reagent in one together. These reactions were reported to be acid catalyzed. So we looked at different catalysts as well. And a second route as well is the chloramine B route, where chloramine acts as an oxidation agent and as well as a potassium salt, for example, as an iodine source. So these are the two schemas we optimize simultaneously. So either we do NES, a catalyst and solvent, the catalyst can be present or not present, or we do a chloramine or iodine salt and the solvent, and both reactions were run for a fixed 2 hours at a set temperature. So we optimize not only one substrate, but 4 substrates altogether. So the chlorinated ortho and para substituted alkene, and as well as the ortho and para substituted alkene as well. So with this, we tested 3 different optimizers from Atinary. Later, Daniel will talk about more which optimizers they are and which of the technology they use below. We did this to see the robustness of our approach, if all the 3 optimizers will find the same optimal reaction condition, or each of them will go to a different reaction condition that is optimal. So how did we consider an optimization to be complete the campaign? So what do we actually optimize for, so to speak? So we looked at 2 things. So the conversion is basically how much starting material remains after 2 hours of reaction time. So how much of the material that we added is consumed. This doesn't need to be selective. So we also looked at the yield, which is basically how much product is formed after 2 hours. So, and we combined these 2 into so-called merit. So the merit is consisting of a factor of 90% yield and 10% conversion. And you may now ask, why did we not choose just yield? Because we wanted, in the beginning, we wanted to favor reactions that actually show some reactivity towards our starting material, even though maybe they are not selective, but we thought this will focus the optimization campaign more towards reaction that actually show some reactivity rather than non-reactive reactions. So when do we stop the reaction? Is basically when the alkenes conversions reached 80%. So we stopped the optimization campaign if all the 4 alkenes, all our 4 starting materials, reach a conversion above 80%. And how do we determine the concentration? We used chromatogram, so the DID signal, and we did with an external calibration curve. We see here the different peak of, for example, the internal standard, the starting material, and the product. And based on these areas, we can then determine the amount of starting material still present, because we know the initial amount we added is always the same, so we know how much product is formed and so on. So then the next question was, how do we actually combine this synthesis robot to an optimizer? And here we go a bit more into the software engineering room, but we already had the IBM RxM platform and the Atinary ST-Lab experimental planning software. So the question is how we connect them. So the RxM platform was always already connected to the robot, as mentioned before. So it was more about how to combine the RxM with the ST-Labs. And we do this by using a backend application, which is nothing more than a little service running in the cloud that queries the reaction ST-Labs platform. If a new set of reactions parameters are available, it translates them into robotic actions that actually can be executed on the platform, as well as it serves a front-end web application, so a little website for the operator to show which chemicals he needs to add into the robot for which reaction. And also this front-end application allows the start of the reaction. So if the operator press start on the website, it actually sends a signal to IBM RxM that will then send the actual instruction to the robot, which executes the reaction. when in the end of the reaction, we have this chromatogram shown before. Based on the areas, we send back the areas to the backend. The backend has the calibration curves, and so therefore it can determine the yield and conversion, which are then returned to the next SDK back to Atinary ST Labs. And this is the cycle we mentioned before. It starts then over after one reaction set, and the Atinary ST Lab suggests a new set of parameters. We get a new set of parameter. Again, we translate them in robotic action. We trigger the start of the reaction. The reaction is run on the robot, which then gives the chromatogram. The chromatogram is sent back to the backend, and so on. So, but the backend application in itself is written in as a Dockerized Fast API container, and I always like the tweet of the creator of Fast API that wanted to apply for a job where he needed 4 years of experience in his framework, but he couldn't apply it since he had only 1.5 years of experience since he created it. So then it uses the RxN for Chemistry Python package to communicate with the RxN side, and the Nexus SDK Python package to communicate with Atinary's ST Labs. So the front end, the web page is written as a Vue.js container. It uses the Carbon design language. You will see it later on in a short video. As well, it's mainly there to display the operator instructions, so which chemicals I needed to load into reactor and so on. All these two containers are then deployed in an OpenShift cluster in the cloud. So the backend, what it actually does is it gets a set of JSON parameters, so these parameters that are queried via API. Then for each of the 3 optimizers we looked at, it gets a batch of 2 recipes. And you may now ask why the batch size of 2. This is due to the fact that we have 2 recipes per 3 optimizers, which equals 6. And if you remember, we had 6 reactors in our ChemSpeed Flex unit. So we can actually run 6 reactions parallel was the initial idea. Then in the next step, these parameters are converted in robotic actions. And what this means in Python code is shown here. It basically appends to a list of actions the robot chronologically goes through. For example, an add action, which is then the material, methanol, and the quantity as a liquid, because it's milliliter, 50 milliliters, and so on. And the quantity of solvents was always adjusted to have a constant reactor volume of 20 milliliters, so that the concentration effects are not there. So as a third step, all these instructions and so on are stored in the database, so it's consistent. And then again, if we choose to trigger the reaction, the backend actually sends the signal to the RxN side to start it as well. It can be used to send back the results. The front end, on the other hand, is a short video. I can play it here. I hope you see it. You can log yourself in, so nobody else could start reactions without me knowing. So after logging in, we see all the parameters. So for example, we see here a list of different reactions and solvents, as well as other information. Then if we go into one of these reactions in more detail that was not run yet, we see these different actions that were basically added in this list shown before. We see here adding 18 milliliters of the solvent, dioxane. Then again, adding the starting material, and here we see a stir action, so just stirring at 45 degrees. Yeah. If you click on Show Operator Instructions, so now we see all the chemicals that need to be present in the reactor in a human interpretable way, so no longer equivalents, but actually the amounts like milligrams and milliliters and so on. So the cool thing is now, if you actually go on RxN, we see the same recipe. You can disregard the chemical formulas because they are not valid, but you see here actually the actions, they are displaying the correct things that we want, the steps we show before. And it ends always with analysis, so we actually know what we do. So if we press Start Synthesis, it wants a confirmation, and if we confirm, it actually triggers the reaction. This always takes a couple of seconds because it needs to validate if all the chemicals are present in the robot. let's see, if we refresh, we see the reaction is actually running. If you go on RxN, we see again this one synthesis execution in progress. This means that it's actually physically running on the hardware. And we see here it started adding the dioxane already, with also a time estimation. So what were some challenges we faced along the way? So first of all, we wanted, in the initial thing, we wanted some MS active ingredients, because MS active ingredients, we see way easier the amounts in the MS signals. So we wouldn't have to do an external calibration curve. But then we noticed that they are not MS active. So we had to do this external calibration curve. And during this calibration curve, we noticed some inconsistency during injecting the same sample. So we injected the same sample over and over, but recorded different chromatograms. And this is not good. And we noticed then after a lot of debugging that it's actually caused by wear and tear on the injection port, because we injected a lot of liquids during this initial calibration. The plastic got a bit bent, and sometimes air was injected. And this was the cause that we then added an internal standard, which we know how much the peak area should be. So we were always sure if the injection was done right or not. This is this blue line. We choose acetophenone, which was added after completion of the reaction. So it didn't react at all with all the other ingredients. So a second problem we faced is we initially had the solvent toluene included in our grid of parameters, but toluene, due to its chemical similar nature with one of the starting material, eluted at the same time in the chromatogram. So we had overlapping peak areas, which we then decided to exclude toluene. A bit the problem was that then the toluene had residues still in the robot. This took a while to clean, but in the end, we got rid of the toluene overall. A third problem we faced is initially we had a third route. So we had 3 routes planned that used DIB. And DIB actually, the side product after the reaction is iodobenzene. And iodobenzene unfortunately had a similar elution time than our products. So we again had these overlapping peaks that we then decided to exclude the route using DIB. And then finally it worked. So I can hand over to Daniel that talks about more about the results of our work.
Super. Thanks so much for your presentation about this challenging robotic experimental setup. Now let me just share my screen. So okay, now share. Perfect. Great. So as Oliver was saying, finally it worked. We have this set of parameters that we want to optimize. We have a well-defined grid with 12,036 points. And here is a depiction of basically the reaction that we want to optimize and the 2 possible pathways, again, using either NIS or using the chloramine B route. And we have 4 different substrates. So now let's see how we can use SD Labs to optimize these chemical reaction routes. Here we use SD Labs proprietary algorithms, which are called Falcon, and we have different flavors of Bayesian optimization. Each of these algorithms varies in their surrogate model. For the interest of time, we will focus on the performance of just one of these algorithms, but they all had similar performance. So the first algorithm that we use to optimize this reaction is called Falcon Light, which is a lightweight ensemble average model. And with this algorithm, we were able to reach the optimal conditions for the 4 substrates in only 23 reactions. Here is how we achieved those results, depicted as a progress chart. The y-axis of this plot shows the conversion, which is one of the objectives that we wanted to maximize. And actually, the dashed line corresponds to the stopping condition. So once we surpass this limit, we stop optimizing the experiments with that substrate, and then we continue optimizing the other substrates. The x-axis shows the experiment number. So when was this experiment carried out? And the shape of the markers correspond to which substrate was used. Note as well the background or the intercalating shades of the background of this plot, which corresponds to the iteration. So here we used an initial set of 11 experiments. And then later on, we used batches of 2 experiments at a time. What we can see here is that the conversion increases over time. This is what we want. And we see that the first experiment that converged at the number 17. So we see that the experiment number 17 was 2-ethanyltoluene. So the algorithm found these optimal conditions and quickly was able to converge the remaining substrates. We had similar behavior for our second algorithm called DNGO, which is a deep network for global optimization. This is a Bayesian neural net. And for this algorithm, it required 25 reactions to optimize all these 4 substrates. Our third algorithm is a Gaussian process Bayesian optimizer. And with this algorithm, we were able to converge after 23 reactions, showcasing how using different flavors of Bayesian optimization, we can accelerate this R&D search using AI. Now, we have seen a bit, okay, how quickly can we reach this target? But we would also like to understand, if there is a common optimal condition, how different were these algorithms in terms of finding that? And that's what we will try to answer in the next slides. So using Atinary SD Labs, we were able to depict these multiple parameters, this highly dimensional space, in a parallel coordinates plot, which you can see now. And the idea of this plot is that then we can visualize trends in multiple dimensions. And the key feature of this plot is that each experiment, each of the conditions that were evaluated would correspond to an individual line. And basically, the columns here correspond to, on the left-hand side, parameters, such as the alkyne group, the amount of chloramine, the iodine source, etc. And on the right-hand side, we see the objectives that we want to optimize, both the yield and the conversion, which are both combined into the single merits variable. And what we are looking at here is the top performing experiments across the 3 optimization campaigns. So we see that we can already start seeing some global trends. For example, we can see that most of the experiments, most of the lines don't use any catalyst. You see, most of them go through none. Additionally, we can see that most of these traces use these polar solvents, methanol, acetonitrile, and acetonitrile with water. And lastly, we can see as well how most of these lines actually don't pass through NIS. They seem to favor the chloramine B route. So here we can already see how these algorithms basically were all able to find fairly similar optimization conditions, but we still see some challenges with this plot. For example, we see that many of these lines are overlapping, and this makes it a bit difficult to find more interactions between these parameters. And in this work, we developed a novel visualization technique to be able to capture these relationships as well with the relationships with the objectives. And here I will show you basically the initiative or the idea behind it. And we were inspired in this kind of visualization because we live in Switzerland. So I know you're aware of the landscape that we have here in Switzerland with many mountains and many valleys. And actually here we are used to looking at contour plots that show the latitude, longitude, and altitude. And we were inspired by this idea, but we wanted to translate this concept to the chemical landscape. And basically here we see the same concept, but looking at 2 dimensions, 2 parameters, for example, the temperature and the chloramine equivalent. And we can see how the altitude here would correspond to the objectives that we want to optimize. So here we see that at a combination of temperature and chloramine equivalent, we have some optimal conditions. So this was the idea of what we wanted to visualize. And you can already imagine that this works in 2D, but how can you do this in an 8D problem, such as is the case here? For that, we needed to resort to a tool called GSNE, which is a dimensionality reduction technique. And what it will do is that it will capture similarities in the high dimensional space, and it will reflect those similarities in the low dimensional space, such that close by markers are similar to each other. So in such a visualization, we can still capture these high-level interactions between the parameters and still visualize them in a 2D setting. So now I will show you how that looks like. So here is the similar plot, but looking now at 8 dimensions that have been projected to the TSNE coordinates. So now the x-axis and the y-axis maybe don't have a physical meaning anymore, but again, close by markers are similar to each other. And just for clarification purposes, we added some annotations, including the iodine source as the first element of the marker of the annotation. We included the solvent and we included the temperature, just to have an idea of how this TSNE algorithm clustered the experiments based on these parameters. Secondly, the color code corresponds to the performance of the experiment. So lighter yellow would mean that the experiment was better in terms of its conversion and yield, and darker purple would mean that the experiment was not so good. So here we can already start analyzing some trends. For example, we see that the plot seems to be divided in 2 regions, and this yellow region on the right and this purple region on the left. And we can see that actually the algorithms seem to find these common optimality regions, mainly 2 of them. One at the top right, using methanol as a solvent, and one in the middle using acetonitrile as the solvent, and actually low temperatures. So we can better understand this by looking at some behavior that we found. For example, we found that the temperature seemed to be organized in this diagonal manner, where low temperatures appear at the bottom right and high temperatures appear at the top left. Next, we can also see how the solvents were clustered. We can see these vertical bins for the DMSO solvent, this vertical bin for the acetonitrile solvent or the acetonitrile with water, and the last vertical bin with methanol. So this kind of plot allows us to see interactions between the solvent, the temperature, and the iodine source. And just to remind you, this plot actually shows the merit as the color code, but the merit is the weighted sum of both the yield and the conversion, showed now here to the right. And we can see how this merit is able to capture the regions at which both the yield and the conversion are high, but not in other regions where perhaps only the conversion is high, such as this star on the left-hand side, where the yield actually is not so good. And likewise, for this single NIS experiment that converged, we see that the conversion is high, but the yield is low. So this better gives us an understanding of the chemical landscape of the problem. We can also use this plot to visualize the trajectory of our optimization. So here we have the same visualization as before, but we added an additional annotation to our markers, which corresponds to the iteration at which the experiment appears. So here we see that at the initial iteration, the experiments are fairly scattered around the space. So all the markers that have a 0 on top would be the first iteration. But then we can see how the algorithm was able to find the optimality conditions or close to optimality conditions within the methanol area here in the right. And further, it will then explore a little bit because it hasn't seen other parts of the space. But as soon as it finds that those parts of the space do not yield to good results, it goes back to methanol and it further exploits around this region. And this visualization allows us to see this trajectory very clearly. Visualization also allows us to evaluate the predictive performance of our algorithm. So before I showed you how these markers were visited by the algorithm, but those markers that don't have a annotation were markers that the algorithm didn't see. So we are only showing here the predicted merits for those. And actually, it's interesting because we can see that for the most part, the algorithm correctly predicts the value. So the color codes seem to match. But there is a region, for example, the central region with acetonitrile, while the algorithm, because of its high uncertainty, perhaps doesn't match as much in its predictions. So this visualization allows us to gain understanding not only of our parameter space, but also of how the algorithm performs. We want to mention that all of these tools are available under the SD Labs platform. These top 3 were the proprietary algorithms developed in this work, but Atinary offers more than 10 algorithms. And also the visualization tools are available under the SD Labs Analytics platform, including many more, as you can see in this screenshot. So we invite you to try this out. With this, we want to close the webinar by saying that we were able to close the loop, establishing an integrated and autonomous workflow, combining robotic automation with RoboRxN and IBM Research, and Bayesian optimization techniques with SD Labs to deploy a fully connected self-driving lab, optimizing constrained chemical reactions of 2 different routes, 8-dimensional space, giving up to 12,000 possible combinations, and achieving a conversion rate of over 80% for all the 4 substrates in 23 experiments across different algorithms as well, covering around 0.2% of the combinatorial space. Here we have a picture of the co-authors, so we would like to thank as well everyone involved in this project, mainly Loic, Fede in this picture. This is actually here at Biopol, at our lab, and in the back we can see our small prototype robot called Mixmax. It's not as impressive as the RoboRxN, but yes, we are very happy for this joint collaboration. And with that, we would like to thank you very much and open the QA session.
Thank you very much, Daniel. Thank you very much, Oliver, for this very great presentation. I would like to draw your attention on the 2 QR codes here. The one on the left will bring you directly to the paper, and the one on the right will bring you to the Atinary website. So I suggest that we proceed now with the questions that were asked in the chat. So let me stop sharing. And we have already a few questions here. The first one is, why did you choose the reaction time to be 2 hours? And why not optimize to reduce time? And there is a follow-up to this. How would you go about optimizing the reaction time? I guess, Oliver, this is for you.
Yes. You can hear me, I assume, again.
Yes.
So mainly the limitation to 2 hours was that we fixed the reaction time was because we had 1 arm on our robot. So if the arm would be occupied, for example, sampling a little sample for the analytics, this would mean the arm would be occupied for quenching the reaction. So we could not ensure that actually, let's say, if we want to do a reaction time between 1 hour 45 and 2 hours, that the arm was free basically to quench the reaction in time to ensure the right reaction time. So we fixed the reaction time to 2 hours. And why 2 hours? It's because it was a lower value than reported in literature in general. So we wanted to optimize basically the reaction as fast as possible so we can run many reactions in a short period of time. And I think different robotic setup allow for easier control of time as a variable, but in our way, we just settled to have it as a constant instead of a variable to optimize for.
Thank you. Thank you very much, Oliver. Here, there's another one. I see the RoboRxN prompts the user with the required amount of reagent, etc. But is it also capturing the actual amount added?
Okay, this is for me again, I assume, and I think maybe it was not fully clear, but the idea is not that we have to add, let's say if it's asked for 18 milliliter, we don't need to provide 18 milliliters exactly. We just need to be sure that at least 18 milliliters are present. The amount that is transferred is then done by the robots for the liquids at least. So the liquid injection is accurate to 0.1 milliliter, I believe, but don't quote me on this. But so therefore, it's relatively accurate enough, I would say, or at least better than my performance with syringes. And for the solids, we had to create little ampules, which we weighted against each other. But they are sure there are some measurement errors, but we also have to highlight that the amounts of solid ingredients are usually not in milligrams per se, but in equivalents. So we discretize these parameters. So there's only, let's say, 1.5 or 2 equivalents of iodine source. So even if there's a small variety between the different samples of 2 equivalence of iodine, it's not so big compared to the 1.5, so on.
Thank you. Then there is a question more for the machine learning side. So the question is, can I transfer the data or the topology to similar chemical reactions? Maybe Danielle, if you want to comment on this one.
Sure. Yeah. So actually, at SD Labs, we have an algorithm that specializes on this topic of transfer learning. It's called SEMOPT, and basically, it will use different kind of neural networks, such as neural processes, to basically transfer reactions that are similar to each other. And this algorithm is actually available under SD Labs. And there's also an associated publication where we show some use cases in the chemistry fields. So yes, this would be a great next step for this kind of reaction to be able to transfer similar reactions. Thank you.
Thank you. Then another one, I guess, for you, Oliver, is what kind of data is collected during the experiments and how is it analyzed?
So in general, I would say the data in itself was mainly the chromatogram that was collected, so the HPLC measurement with the DID sensor and the MS according. But we mainly focus on analyzing this. Let's say the temperature control and so on was usually nearly perfect because it's industrial hardware, so there was not so much fluctuation in between this, as well as the liquid, as mentioned before, the amounts of liquids that were added to the reactors. They should be fairly constant between the different experiments in terms of variance. So there, sure, we had some measuring errors with the used up injection port, but this we noticed through the internal standard. So before we reported any of the collected reactions, we always checked if the internal standard was actually the right amount. So if this was not the case, we repeated the experiment. If it was the case, we reported the experimental yields and conversion back to Atinary.
Thank you, Oliver. Here, another one for, I guess, you, Danielle, is about the algorithm. So why did you choose these three specific optimizers and not the other one that are also available through the platform?
Yeah, so we chose these algorithms because one key thing that sets them apart is the ability to create this very constrained space, which was the case here. So as you saw, we have two routes. Each route would feature different kind of chemicals and different conditions. On top of that, we had several constraints on the temperatures, for example, to not surpass the boiling point of multiple solvents. And we found it very difficult to set this up on, let's say, other out-of-the-box solutions. So that's why we decided to go with the proprietary algorithms from SD Labs.
Thank you. Then the question, I guess, for you two, in slide number 19, you mentioned that there are 11 shared reaction as starting point. Can you explain how those were selected? And can you clarify, is they're already included in the total number of experiments, 23 in total for Falcon Light?
Should I go? Yes.
I think.
Yeah. So these 11 reactions were actually chosen, uniformly across the constrained space. And we wanted to maximize the diversity of these samples to ensure maximum coverage in these initial 11 reactions. And then what was the second question?
It is, if those 11 are already counted in the total number of experiments.
Yes, so those 11 experiments are already included in the 23 experiments. So we did 11 at the beginning and then followed up with 2 experiments at a time, totaling 6 loops in total and 23 experiments.
Thank you, Danielle. And maybe a last one that probably is for you, Oliver, with the challenges that you reported, is what is the one thing you would like to redo and make it better?
I think personally, because I spent a lot of time in the lab, so I would say the analysis part, until we figured it out, was a lot of work. And I think there's also a related question to this: why not use the GC to analyze? And this is the simple fact that we didn't have a GC on hand that we could use. If I prefer, I would use a GC over an HPLC for this setup, definitely. But as so often, sometimes you don't have the lab resources to go out and buy a GC. And what I would do different, I think, selecting more reagents that are actually liquid, because it's so much easier to deal with than making solids solid handling in general, I would say, is a bit trickier with robotic hardware than liquid dispensing.
Thank you very much. As this webinar is reaching to an end, I'd like to thank you all for your participation. We hope you found this webinar informative, and we cannot wait to see you again at future events. We encourage you to follow us on LinkedIn and Twitter, and to sign up for our newsletters. Please do get in touch with us if you have any question. I wish you all a wonderful day. Oliver, thank you again for your participation, and Danielle as well.
Thanks
Thank
for
you
having
very
me.
much.
And thanks everybody for listening.
Thank you, Danielle.
Thank you all.
Have a great day. Goodbye.